Good morning, my dear students. Today we discuss about the staining technique in the microbiology or differential staining techniques in the microbiology. This is one of the most important technique by which you can apply it for detection of the microorganism. There is a bacteria, basically which type of bacteria that can be identified by this mechanism or by this test. So after invading the microorganism or the microorganism is outside, that can be identified by this detection method. Let us start. The bacteria is the microscopic organism. They are also for the most part, and in order to visualize them, to study the structure, shape and other characteristics, it becomes necessary to make them more easily visible, and that means the contrast with their environment, so they can easily stain. And stain is the procedure or use some dye to identify the microorganism. Color dead cell organism. Types of dye: acidic. Their negatively charged acidic radical impact color in the fuchsin, acid fuchsin, magenta green and neoglycerin in Indian ink. Basic color: positively basic radical combines with the negatively charged particle in the cytoplasm and gives color. As for example, hematoxyl, methylene blue, crystal violet, Jensen violet. And neutral, both the positive as well as the negative charged impart a different color to different cell components. As for example, Jensen stain, Leishmanian stain and Gronheit stain. Staining method. There are different staining method. One is the positive staining, another is the negative staining. Where the actual cell are themselves color or appear in the clear background, that is called the positive staining. And positive staining can be again classified into two groups. One is the simple staining, another is the differential staining method. Simple staining is stain which provides color contrast but gives same color to the bacteria and the cell. As for example, Loford, methylene blue, polychrome methylene blue and dilute carbol fixture. Differential staining is stain which impact different color to different bacteria is called the differential stain, which contain more than one stain. As for example, the gram stain, acid fast stain and the special stain. Negative staining, where the stain remain clear and the background or color uncolored and the background is colored, to create a contrast to aid the better visualization of the image. So there is a difference between the negative staining and the positive staining. In the negative staining, the background is the color, but in case of positive staining, the background is uncolored, but the organism is colored. So in the negative staining, there is a Indian and nigrosin are used for the coloring agent or drying agent. Bacterial smear preparation. Before staining, we first to prepare a smear, and in that case you take two slides. In the distribution of bacterial cell are slide for the purpose of viewing the under the microscope. What is the methodology? Typically aseptically, that means those where the smear will be prepared, that room must be aseptic condition. That means they are totally free from the microorganism for to prevent the contamination. Aseptically, small sample of the culture is spread over slide over surface. This is allowed to air to dry, or in the heat, mild heat it is applied to dry. And the next step, heat fixation to help the cell adhere to the slide surface, and the smear is now ready for staining. And a thin film will be formed by the apply by the another slides. So this is the procedure by which you can apply it. Basically, first of all, you take one slide washed by the alcoholic solution, then dry it, then one loop full of microorganism culture is placed over the slide, then another slide just spread over the slides and to form a thin film just in the position number 4 or last slide, and then it will be kept for the dry air, and after it will be fixation by the heating applied in the Bunsen burner just below the slide to take over 2 to 3 times. The section being embedded in the paraffin and the necessary to remove the paraffin to water stain and protection, and the paraffin is first fixed. This is not smear fixation by the heat fixation. Passes air dried smear through the flame 2 to 3 times and do not overheat it. Allow slide to cool before staining. Another is the methanol fixation. Methanol is a volatile liquids. If the methanol is used, it will be removed easily and automatically, and then it will be fixed. Place the air dried steam in the colaphane and just the methanol 1 minute, alternatively floods, and the stained slide allowed to dry before the staining. First of all, simple staining. The low-fat methylene blue. It is generally the most useful and so that characteristic morphology or polymorphs and lymphocytes and other cells can be clearly than the stronger stain such as gram stain or dilute carbon fusion. Polychrome methylene blue. This is made by allowing the low-fat methylene blue to ripen slowly, and the slow oxidation and methylene blue form a violet components, and that gives a stain in the polychrome components. The ripen takes the 12 months and more complete, and it will be ripened quickly. Addition 1% of potassium carbonate to the stain, and it is always embodied the MC Federin reaction, and in contrast to the staining of the such structure by the methylene blue. And the violet component stain will be structure red color, and the acidic capsule material of anthrax bacillus is the MD Frieden reaction, dilute carbon fusion, which is used for the Nielsen stain. What is the requirement of this particular for staining? Number 1, low-fat methylene blue, dilute carbon fusion, distilled water, compound microscope, cedarwood oil, and fixed smear. Procedure: take a smear and the slide, the heat fixation and passing them 2 to 3 times, and a carbon fusion, and gram will be smear dilute. So in case of simple staining low-fat methylene blue, the violet color will be produced. You can see under the microscope the gram staining. Another is the gram staining. This is the most important for your syllabus. One is the gram staining, the gram positive and gram negative. And gram staining is most widely used for the differential staining in the microbiology. Gram staining differentiates the bacteria into 2 groups. One is the gram positive bacteria and gram negative, like Staphylococcus aureus, gonococci are the gram negative, or E. coli. This is the short history. Hans Christian Gram, which discovered the gram staining. The gram stain was discovered, and the Danish physician Hans Christian Gram was working in the Berlin in the 1883. He later published on the procedure in the 1884. At that time, Dr. John Gram was studying lungs tissue section from the patient who had died of pneumonia. So this is the miracle. So is the most important invention or detection of the microorganism by the very simple technique, that is gram staining by the Christian Gram. So thankful to me, Dr. Grams. Original formulation of the gram staining: the aniline and Jensen violet, Lugol solution, that means iodine solution, absolute alcohol, and Bismarck brown. At the 1845 and 1940, the German scientist Franklin added the final steps of the staining. Only in case of gram staining, the gram Christian Gram, only in that case only the gram positive organism can be identified, but the gram negative organism cannot be identified. But then this will be overcome by this another method. Then used another dye, safranin dye, and Streptococcus bacillus, Streptococci. So gram-negative positive bacteria can be identified only adding the safranin. So this is the modification of the addition of this basic principle of the gram-positive bacteria. Basic staining procedure. Basically, the gram reaction is dependent on the permeability of the bacterial cell wall. Actually, it's dependent on the cell wall structure, and cell wall structure is basically composed by the peptidoglycan. In case of gram positive and the gram negative, but the component varies from gram positive to gram negative, and the reaction material, the coloring material react with the different cell wall of the bacteria. The cytoplasmic membrane to the di-iodine complex. The gram-positive bacteria in the crystal violet di-iodine complex combine to form a larger molecule which is the precipitate within the cell. Also, the alcohol and acetone mixture, generally 1:1:1 ratio, which are decolorizing agent, causes dehydration of the multicolored peptidoglycan of the cell wall. This causes decreasing the space between the molecular causing of the cell wall to trap the crystal violet iodine complex within the cell. Hence, the gram-positive bacteria do not get the decolorized and return in the primary dye appearing in the violet. Also, the gram-positive bacteria have the more acidic protoplasm, and hence it will be there. So, gram-negative bacteria, actually gram-negative bacteria, basically it is composed by the lipopolysaccharide portion. So, lipid content can be solubilized in presence of the alcohol. So, alcohol dissolve and it will be neutralized. In case of gram-positive bacteria have the thick wall of peptidoglycan, and the others polymers of peptidoglycan constant interweaving flamelets made a alternative N-acetyl muramic acid and N-acetyl glucosamine monomer in the gram-positive bacteria. There is a well trichoic acid. So gram-positive bacteria, peptidoglycan, this is the main chemical component in the thick wall and as well as between the cell wall and cell membrane. So cell membrane trichoic acid. And in case of gram-negative bacteria, there is a outer membrane is the phospholipid and the bacterial lipopolysaccharides. So there is a basic difference between the gram-positive and gram-negative bacteria and the cell wall. So based on this cell wall component, the dye will be react or complex with the dye due to this component outside the thin peptidoglycan layer, and this space between the outer membrane and peptidoglycan is called the periplasmic space. The outer membrane protects the gram-negative bacteria against the penicillin and lysozyme. This is the basic structure of the cell wall of the different, in case of gram-positive, the pillars, allegrams and gram-positive bacteria. These are the cell membrane structure of the gram-negative and in the gram-positive structure. Gram-positive is the thicker and here is the thinner and component of peptide N-acetyl muramic acid and N-acetyl glucosamine, but here is the lipopolysaccharide and the phospholipid. So this is the basic component different, that's why their color react with the different color reaction. So in case of gram-positive bacteria, there are the steps involved in the stainings. The step staining in the first one, the crystal violet solution is the primary dye. Then second step, iodine solution is a moderate, and for the cell staining for purple color and cells remaining the purple color. Second to the alcohol decolorizing, in case of gram-positive it will be written and it will be purple color, but in the step 4 the safranin is used. In case of gram-negative is produced red and remove the violet color. So you can identify if after the final conclusion, if it is violet color the gram-positive, if the red color it is a gram-negative bacteria. Method concept of the 4 components: one is the primary stain, crystal violet, methylene violet and Jensen violet. And moderate gram-negative iodine or really Lugol solution. Lugol solution is nothing but an iodine solution. Decolorizing agent, alcohol acetone or alcohol acetone mixture 1:1 ratio. Counter stain, dilute carbonfuran, safranin and neutral red. And sandy 4 stain or gone for gonococci. Procedure for the step, for the 4th step: primary staining, the smear will be covered with a Jensen violet and 1 minute on wash with water. Moderate, it is covered with a gram iodine, kept for 1 minute and wash with water. Decolorization, the smear is covered with alcohol and it wash with water immediately. Counter stain, the smear is covered with a safranin, kept for 30 seconds and washed with water. And using the filter paper, the slides is generally bottle dry, and the drop of the cedarwood oil and liquid paraffin on the smear. Just the microscope for increase the light by raising the consider and the slide exam under the oil immersion objective using the plane mirror. As a result, bacterial managed to keep the purple color dye only got it is a gram positive. If it is a red color, it is a gram negative. Another procedure for the tissue section, this is not required for at that time. If you can read it, you can. Modification of gram negative. There is some modification since the original procedure of gram may be variant from the gram staining technique has been published, and them involved in the others method, and others include some minor technique variant for no value. Bethel new pointed out and the variation the gram staining procedure of definite shapes and acceptability. The final result is outcome the interaction of this possible variant and various modification of the gram staining procedure like couple of Beeman's modification. Here primary stain is the methyl violet and decoloring the acetone or acetyl alcohol mixture. Jehan's modification, primary stain is the methyl violet, decoloring the absolute alcohol and counter stain for neutral red. And Piston or Morel modification, primary stain is the crystal violet and decoloration. Oyster modification, primary stain is the Jensen violet. The common error in the staining procedure. Common error in the staining procedure, excessive heat during the fixation. Generally, we are the common error during the staining procedure, and you take care of this when you staining procedure done. You take care about this, and you remember in your mind excessive heat during fixation. That means they lost the microorganism, low concentration of crystal violet, excessive washing between the steps, inefficient iodine exposure, prolonged decolorization, or excessive counter stain used to color the negative color. Application of the gram staining. The rapid perform the diagnosis of the disease such as bacterial meningitis, selection of empirical antibiotics based on the gram staining findings, selection of the suitable culture media based on the gram staining findings, screening of the quality of clinical specimens such as the sputum test in case of tuberculosis contain the many past cell and few epithelial cell, counting of the bacteria, apparition, morphology and the type of bacteria in the clinical specimen. Jehan's staining for the acid fast bacilli. Another very important technique. You cannot be identified by the gram staining procedure, but another bacteria which are acid fast bacteria. They are generally produce some acids. Generally they produce mycolic acid by the some mycobacterium species like Mycobacterium tuberculosis and Mycobacterium leprosi, and they are and Nocardia also. The acid fast bacteria modified by the Paul Ehrlich, which is the father of chemotherapy. He developed this method how to identify the Mycobacterium tuberculosis or lepra or nocardiasis. They produce some acid and that can be identified. The principle of the basic Jehan's stain by the acid fast fustian. Generally the procedure is the same, but only the difference the carbol fustian is used for staining procedure, and this is the staining method applied for the help for the drive for the penetration of the tubercle bacilli, and once stained it cannot be easily removed, and the tubercle bacilli resist the decolorizing agent of the acid fast alcohol, which is convert the acid fastness of the bacteria, and the others microorganism which are easily decolorized the acid alcohol and consider the non-acid fast bacteria. So on the acid fast bacteria, it is absorbed and color of the counter stain is the blue, appearing blue, when the acid fast cell written in the red color of the primary stain. There is some ABS modification, Nehal Jensen hot stain, and the acid fast staining basic fustian, primary modern stain in the strong carbol fustian or it is a red color, or decolorizing agent is the acid alcohol. So 3% alcohol and 5% and constant in the methylene blue. So in bacteria it will be produced red, and non-acid fast it will produce blue color. So you can identify whether the organism it is a acid fast or non-acid fast. If the color, final color will be produced the red, it is acid fast after carbon fusion is added. If it is blue color, that is non-acid fast procedure. It is a short procedure to make a smear, dry air and fixation. Blood smear will be carbon fusion stain and steam will the 5 minutes. Add more the carbon fusion stain as needed. Cool slide for 5 minutes and wash with the distilled water. Flood slide with a acid alcohol for 15 seconds. After it will be color change. If it is red color, acid fast, no red color, there is a and tiles of the sorry 45 degree sink and the alcohol is drop wash and the range with the distilled water. Alcohol loafens methylene blue stain, leaves loafels blue stains on the smear for 15 to 20 seconds and range in the slides. It's the dry for the oil immersion object view. Microscopy reading the stain of the smear contain the pink colored cylinder rod shaped structure has been carved in the acid fast bacilli seen among the color or multicolor plus cell and the smear is the positive for the acid fast bacilli. Differential modification of the acid fast staining, generally 5% sulfuric acid or 1% sulfuric acid for the decolorizing agent and 0.2% sulfuric acid is the decoloring agent for the staining for spores. Fluorochromes staining, this is the another staining by the fluorescence microscope and generally the 250 on 50 magnification is rapidly generally a very good technique and you can identify the most of it, all of the microorganisms by the fluorescent staining. This is the procedure by which you can identify the place the dried and fix the smear and staining the clock and the sputum should be thin covered with a smear and aluminum phenol and levo stain are used to temperature for 10 minutes and wash and stain with the water and covered with a 1% of solution on 1 minute. Wash a decolorizing agent. So these are the procedure you can identify by this procedure and fluorescent stain microscopy a sputum smear stain with the uranium meramine. Fluorescent are the UV microscope is not needed and volatine's content volutine it is another component. It is identified by these following methods. Procedure, just the same procedure you can read it carefully and continue it. Next, Albert microscopy competition for Corynebacterium diphtheria stain can be Albert stain green method. These are the different method by the Corynebacterium diphtheria. They will produce the diphtheria. This Corynebacterium diphtheria showing the Chinese letter and arrangement the angles and each other color and bluish black. Magnetography granules of the cell you identify the Corynebacterium diphtheria. Another is the staining of the spores. The spores are the highly resistant and metabolic inactive forms, but disease can be identified. This morphological of the bacterial endoscopy is best observed under the stain and film and contrast to the microscope, whereas it appear the flagella, refractive and oral and spherical bodies to maintain the others and differential staining to available the staining the spores and the modification of the Gehal Nielsen stain and for weak 0.25% sulfuric acid used for the decolorizing yield to spore and it blue and stain bacteria and lipid granules. So these are the different processes. Green stain for the spores and microscopy. These are the color you can identify spore. Then the capsule contain bacteria and the how to advantage. This is the basic principle and the methodology is given with the both negative and the positive staining method by capsulation. The positive condition of the smear and another is the capsule can be and this is the structure and color contrast you can identify whether it is a capsule or not. Next, another staining for the spirochete. Generally 3 spirochetes in the available in our environment. One is the Triponema palladium which produces syphilis, leptalacia and borrelia. These are the 3 spirochetes and it is identified by the different staining technique already given in these slides. Next is the flagella staining. Flagella also part of the organ of bacteria and how it will be identified there is a that will be described. So thank you. This is all for the staining procedure, different staining procedure by which you can identify the bacteria and to identify the disease also bacteria also. So it is helpful for the treatment also. So thank you. Thank you very much for listening the lecture. If you like it, please like me in your like box, or if any comments, please comment your Google Classroom or in the chat box. Thank you, thank you, thank you again, all of you.